I'm pleased to introduce speakers for Session 3. Session 3 for examples of model integrated bioequivalence for long-acting injectable and implantable drug products focus on best practice development has 3 speakers. In order of presentation, Joga Gobburu, PhD, is a professor in both the School of Pharmacy and the School of Medicine at University of Maryland, Baltimore. Prior to his current role, Dr. Gobburu held various positions at the US FDA from 1998 and 2011. Dr. Gobburu has experience with overseeing the review of numerous investigational new drug applications, new drug and biological licensing applications, FDA guidances and policies pertaining to drug approval and labeling. Géraldine Ayral, PhD, is the VP Application at Simulations Plus, Lixoft Division. Dr. Ayral leads the team in charge of all pre- and post-sales activities, such as demonstrations, trainings, technical support, and research activities, such as improvement and development of new method, application to case studies, publications, as well as the monolith suite development, such as product specification and testing. Pharmacy Gajjar, BA Honors, M.Med., PhD, as a principal scientist at CEDA Pharmaceutical Services, with responsibility for pharmacokinetic and pharmacodynamic modeling. Dr. Gajjar has a strong mathematical background with a BA and MS from the University of Cambridge in UK and a PhD in applied mathematics from the University of Manchester, UK.
Thank you very much for giving me this opportunity to share my views on how to accelerate long-acting injectable generic drug development using a model-based analysis. This research is made possible via a grant from Center for Research on Complex Generics, which is a collaboration between University of Maryland and University of Michigan, funded by the FDA. So we are grateful for this funding, which made this research possible. The work is mostly conducted by Donald Lee, who's one of my graduate students at the Center for Translational Medicine. I wanted to acknowledge his efforts upfront. We are all familiar with several long-acting injectables. They have been on the rise recently 10 to 15 years. And these are some examples of the products that you might have come across. In order for us to get started on understanding how to accelerate long-acting injectable generic drug development, we first undertook a systematic way to gain R&D intelligence on long-actings. So the way we looked at it is we systematically looked at all the approved products by FDA, then looked at Orange Book, then excluded OTC products and discontinued products and such. Then at the end of all this research, what we found was there are 6 generics per brand product of immediate release, okay? But when we look at long-acting injectables, it's much less than 1 generic per brand product. So there is a big discrepancy between the immediate release versus the long-acting injectables, which can be considered as complex generics. And this lack of generics, in my opinion, is a national concern, because if we look at history, diseases like HIV are now under control only because of the availability of generics, because it allows more patients for affording these essential medicines and thereby improving the healthcare in general across the US, across the globe. So that is why not having generics is really a national concern. Now, one might ponder, or it's a good place to start for us, to think about what are the challenges? Why is this the case? So we came up with 3 different reasons that are potential challenges why we see so few long-acting injectable generics. First, that the testing is in patients, the bioequivalence testing, which means recruitment is slow, variability is inflated because these are patients, they may have comorbidities and other drugs on board, and then washout is impossible. Then the long duration has implications in terms of reaching steady state is impractical, because imagine if a drug is to be dosed every 6 months and you need 4 half-lives or so, we're talking about 24 months to reach steady state, and discontinuations are high because of this long duration, and these are patients, right? And when it comes to the trial designs and analysis, they're rigid, in my opinion, which is rich sampling is impractical. Although you might be able to pull off a study for an year or two, having rich samples, you can't make the patients come every week to give a blood sample and such. So rich sampling is impractical, and that means you would not be able to conduct non-compartmental analysis. Then, because of the duration and the nature of the products, parallel design perhaps might be more suitable, but parallel design comes with a cost of larger sample sizes. And typically, because of the complex release characteristics of long-actings, additional bioequivalence endpoints like partial AUCs are also required. That also adds to the burden of demonstrating bioequivalence. And for several of these reasons, conventional bioequivalence analysis is inefficient, right? So these probably are creating a barrier to entry, so to say, for long-actings. Now, what we challenged ourselves with is, can long-acting generic be developed in half the time at half the cost? So perhaps that would accelerate generic drug development. The motivation for us to even fathom such a ambitious goal is this initiative by the FDA to use model integrated evidence to support generic drug development and approvals. So this is a very forward-looking initiative by the agency and their leadership, and we piggybacked on this initiative and applied that to long-actings injectables. So how do we disrupt the generic drug development for LAIs? So I'm going to present to you in 3 parts. The first, I'll present to you the new paradigm, which I'm calling learn apply paradigm, where you learn from an abbreviated bioequivalence trial and you apply those learnings to a full BE study using model-informed, model-integrated bioequivalence testing. Then I'll go through the scientific evidence to support the learn apply paradigm. And finally, I'm going to present to you a development and regulatory strategy that you can consider for your generic development. What is this learn apply paradigm to long-acting generics? It includes 3 steps. It's quite simple. First, conduct an abbreviated BE study, which is shorter study, smaller study, single dose bioequivalence. Then use the ABE data to perform a model-based analysis using population PK modeling, which is a very standard way of analyzing these data, and then use the population PK model to simulate the full bioequivalence study on a computer. That's the model integrated full BE study. So just 3 steps: abbreviated study, model-based analysis, and then model integrated full BE study simulations. Where is the scientific evidence for this? So that is what the grant is about. So we generated scientific evidence. We tested if this is feasible. And the way we tested it is using modeling and simulation techniques. Now, these approaches have been the fundamental basis for all generic policies for the past 30 years. So you look at all the way from single dose versus multiple dose and such, all the policies have been derived using simulation techniques. And we just use the same approaches. This is the research plan. We evaluated various designs of abbreviated BE study, then we subjected them through model-based analysis, and then we simulated the full bioequivalence trial. And I'll go through the scenarios that we tested. So the first where we started was we considered a long-acting that's given every 1 month, and then these are patients. Although I'm saying it's 1 month, in terms of the simulations, it really does not matter because the principles and the inferences, the conclusions we draw here are applicable for 3 months, 6 months, and longer products as well, because we are dealing with truncation of the study as a percentage of the total duration rather than number of hours, really. Okay, you will see it in a minute. So we considered a sample size of 50 per arm, and the sampling ranged from 100%, meaning full, bioequivalence study sampling, to 75%, 50%, and 25%. So imagine if you were to conduct a trial for 4 months, and here 25% would mean that the trial is only for 1 month now, and in only 50 patients. And we considered several scenarios of test reference ratios of 1 through 0.8. I'm going to showing you the 1 and 0.8. Why 1 and 0.8? 1 represents reference versus reference. So if truly the generic is a generic, then it should pass most of the times. And if truly the generic is not a generic, the test product is not a generic at 0.8, then it should fail most of the times to pass BE. We also considered different levels of variability, 10 to 20%. And these are the different scenarios we have evaluated for the abbreviated BE study. So to depict how on an average the concentration profile would look like, what you have on the left-hand side is the 100% trial duration, for example, and 75%, 50%, and 25%. Then the next step is to use modeling to analyze these data, right? So the data shown here are for the 50% truncated for the reference on the top, and the test is on the bottom. And after these data are subjected to modeling, what you see on the right-hand side is the model predicted concentrations as well, going through the data for both test and reference. Then you will have the individual PK parameters for clearance, volume, and you will also be able to derive, because we have the full profile now, the area under the curve, the Cmax, and for all other subsequent calculations, you would be able to derive the individual PK parameters. So we talked about the different scenarios for abbreviated bioequivalence trial. Now we are talking about the modeling of this abbreviated trial. We also wanted to make sure that we have an understanding of the potential bias in terms of estimating these parameters for the different scenarios using modeling. So what you see here is are the parameters: volume of distribution, Cmax, clearance, and AUC. Let's focus on volume distribution and clearance for a minute. What you have on the x-axis is the mean absolute prediction error. 0 would indicate that there's no bias in terms of the predictions by the model, and let's say 10 means that there is an upward bias of 10, or on average a bias of 10%, because it's absolute prediction error in the estimate. So as you can see, the x-axis, the full trial duration of 100% for all the parameters, the bias is pretty nominal, like 2% or 2.5%. But as the trial is truncated, that meaning you look at, you travel right to left, the truncation evidently increases the bias in the estimation of these parameters, particularly the AUC. And you can think about it this way, because the truncation is past this Tmax. Cmax is not that affected, but AUC is affected with look here, the 25% truncation. There is about 10%. 10% bias is not bad, but still there is this trend as expected. So that means that the scientific approach is as expected. The results are in line with the prior expectations. Now, the next step is, now we take the model for each of those scenarios and simulate full bioequivalence trial and then conduct the NCA. Now, the full B trial will have 100% sampling, no truncation, and we also change the samples, increase the sample size to 200 per arm compared to the 50 from the abbreviated. That's 4 times larger sample size. Okay. Now let's look at the 2 most important aspects of the full bioequivalence trial performance. One is the type 1 error rate, meaning we do not want to approve a test product as a generic when it is not. That is the left-hand side panel here. As you can see, at all, irrespective of the trial duration or the truncation, 25% to 100%, the alpha is preserved at a very low level, as you can see from the left-hand side panels. But when it comes to, and at both low and high variability, okay? Now, when it comes to reference versus reference, meaning can we identify, these are the true positives, basically. So there you see that it is 100%. Or almost 100%, which is reassuring that the learn-apply paradigm to long-acting generic drug development is preserving the type 1 error and a high power for the reference versus reference case. Then I want to now bring your attention to a recent draft guidance on pharmacokinetic-based criteria for supporting alternative dosing regimens for a cancer indication. Now, you might wonder, what does it have any relevance to this? The relevance is the principles alluded to, described in this guidance are outstanding, simply outstanding. I think this is my new favorite guidance, which really shows the how seasoned the regulators are. And I really highly recommend that all of us review this very carefully. Let me highlight one particular aspect that is shown here. A PK-based approach relying on POP-PK modeling and simulation can be applied to support the approval of alternative dosing regimens and such, right? So, but the principle of comparing alternative dosing regimens is the same principle of comparing pharmacokinetics in terms of a generic versus test. The principles are the same. So, and particularly this guidance says simulations can be performed to derive PK profiles and parameters following these alternative dosing regimens. Hence, I'm saying you can use the same approach to deriving the full bioequivalence parameters using simulations. So I hope you see that the approach is consistent with the regulatory policies and what is accepted by the FDA in general. There is more work to be done. First, evaluation of model qualification methodology has to be elucidated, meaning that how would a company or what would a company need to bring forward to validate the model that is to be used to simulate the full bioequivalence trial. There has to be a consensus about that with the agency, but what type of a qualification methodology would be most appropriate? We have done that, but I only have 20 minutes here, so I'm not able to show that, but we will be sharing that at a different forum. Then evaluation of different model estimation methods. So what you have seen here is maximum likelihood estimation, but what about Bayesian estimation, and what about other expectation maximization based algorithms? Then the last one for the next steps in our journey is expansion to more complex absorption products. So what we have chosen here is a relatively simpler profile, but once you go into the long actings for some of these anti-psychotic drugs, the absorption profile, the release profile is a little bit complex. So can we still apply the same approach to such absorption profiles is what we'll also look into. So to briefly summarize, that learn apply paradigm to LAI generic drug development involves 3 simple steps: conduct an abbreviated BE study, analyze using modeling, then use the model and the parameters to simulate a full BE study. That's it. How do we implement it in drug development? So if you are a generic company, then what you would do is you would have the product development. You will still probably use some kind of quantitative approaches to find, based on in vitro, in vivo analysis and such, come up with a product. Let's say you have that product that you're confident to go for a bioequivalence trial. You will develop a model integrated BE plan. So you will have to do some of the simulations that I've shown you in this plan, and then you would submit it to the FDA for FDA agreement. And once you have the agreement by the FDA in terms of the design of the ABE study and the analysis, model-based analysis of the ABE study and the BE simulations, the full BE simulations, you would go ahead and conduct the ABE trial, apply the model integrated BE approach, and then you file the ANDA. So this is going to be the development regulatory strategy for generics. So thank you very much for the opportunity. And if you have questions or you need any information about this, please feel free to write to me. Thank you very much.
Hi everyone, I will present you a novel model integrated designs for bioequivalence studies of LAI products, embedded as a complete framework with the monolithic suite. I will be quick on the intro as you have already heard this in the previous talks. So LAI are designed to require infrequent dose administration to improve patient adherence. They have extended drug release, leading to flip-flop kinetics and long apparent half-lives. For LAI, the typical BE designs are impracticable. Parallel designs, for instance, have a low power because of the high variability between individuals. Let's take an example to check this in more detail. Among the LAI on the market, I will focus on Buprenorphine, which you may know as Sublocade. It is used for opioid use disorder with a monthly dosing regimen. The draft guidance on buprenorphine extended release suggests a single-dose parallel designs for bioequivalence trials. The three metrics to look at are Cmax, AUC from 0 to T-last, and a partial AUC from 3 to 4 weeks. If we simulate many parallel design BE trials using the published population PK model for buprenorphine long-acting injectable, assuming a 5% difference between the test and reference formulation, we can plot the power of the trial versus the sample size. We see that to achieve an at least 80% power for the three metrics together, AUC 4 to 5 weeks will be limiting, and we need at least 500 individuals, so 250 per arms. So indeed, for a parallel design for buprenorphine, the power would be pretty low. Alternatively, we could consider a crossover design. Because of the long apparent half-life, we need a long washout period, and this leads to long trials and therefore high dropout rates. Again, we can ask ourselves how long exactly. To choose the washout duration, the FDA guidance indicates to use more than 5 half-lives. The apparent half-life for buprenorphine is around 73 days, so 73 times 5 would mean a washout period duration of 1 year. Another criteria is that if the pre-dose residual concentration is above the Cmax of that individual, the subject's data cannot be used. So again, we can simulate from the buprenorphine model and check the percentage of individuals having a residual concentration below 5% of Cmax for several durations between the two doses. I have plotted this percentage of subjects with residual concentration below 5% of Cmax versus the duration between the two doses. If we want to be able to keep at least 90% of the individuals, we need 52 weeks, so 1 year between the two doses. So at the end, that would be a quite long study duration. These challenges have prevented the development of generics for long-acting injectables. And this is why we need to propose alternative designs. And designs incorporating model-based evidence could be a solution. The alternative design we have proposed and investigated is the following. It is a 2-period, 2-treatment, 1-sequence crossover design with no or limited washout. The data for one individual could look like this, for instance, with the first dose of reference, followed by the second dose of the test formulation, before the amount of the first dose has been completely eliminated. So the data for the second period is a combination of the first and second dose, and we need to correct this. To do so, we will use the published population PK model for the reference formulation and estimate the individual parameters for the data of the first period. Using these estimated individual parameters, we can predict the carryover concentration of the reference dose into the second period. This prediction can then be subtracted from the data of the second period. This way, we have separated the contributions of the reference and test dose, and our corrected data is as if we would have had a long washout period. So we can apply the usual bioequivalent statistical analysis for crossover designs and get a BE conclusion. To be able to apply this procedure, there are a few requirements. First, we have a single dose design, so it must be possible to give the long-acting injectables to healthy volunteers. Second, the subtraction that we are doing here means that we are using the superposition principle, so the PK needs to be linear. And finally, we need to have a population PK model characterizing the reference formulation as we are using here. All the steps I have explained here can be done using the applications of the monolith suite, so I will show them to you using an example dataset. I here have a dataset for a BE trial with a reduced crossover design, and on the other side I have a monoX project where I have implemented the model for buprenorphine. So I have here the structural model, and here I have put the population parameter values for the reference formulation that I have fixed. To create the monoX project, I had to set up a data set. So for the moment, it's just a dummy data set. And what we're going to do next is to select our B trial dataset. I set it up and we can have a quick look at it. So we see that if I stratify and color by formulation, I have first a dose of the reference formulation and then 4 months later a second dose of the test formulation. The duration between the two doses is more than the usual 1 month interdose interval for buprenorphine, but much less than if I would have waited for the first dose to be completely eliminated before giving the second one. What I need to do next is to estimate the individual parameters corresponding to the data for the first period. What I'm going to do is to go to the Data tab and filter to keep only the lines for which the formulation is equal to reference. I apply and I can check in the plots that I now have only the reference dose. Now I will run the scenario and this will skip the population parameter estimation because anyway we have fixed all population parameter values and estimate the individual parameters. And here I'm calculating both the mean of the conditional distribution of each individual and also the mode, which are the EBEs. In the plots, I can see the individual fits with the estimated individual parameters, either the conditional mode or the conditional mean. I next would like to simulate how these curves would continue if I would not give the second dose. For this, I'm going to use Simulix, so export my Monex project to Simulix. In the Simulation tab, I will simulate the same 20 individuals using the conditional mean estimated by Monolix, the treatment of the B trial dataset that has been transferred from Monolix to Simulix, and I need to create a new output corresponding to the measurement times of the second period. I could type the values here, but as there are quite a lot of them, I'm going to load an external file. When I click Run, I'm going to get the simulations corresponding to the first period, but also the carryover into the second period. These values here, I need to subtract them from the data measured during the second period that are in the original dataset. So to do this subtraction, I have exported these predictions, and then in R, I have a very small script to load the dataset, the simulated value, calculate the difference, and export this as a corrected data.csv file. The last step is then to analyze this corrected dataset into PicAnalytics. Here's my PicAnalytics application. So I start a new project, select the corrected dataset. In the plots, I can have a look at my ref and corrected test data. To set up the bioequivalence, I go to the Tasks tab. In the bioequivalence design, it's detected as crossover. I will have as factor ID and formulation. I don't need Aucs as it is redundant with the formulation. And then I'm going to have as parameters Cmax, AUC last. I don't need AUC to infinity, but I would like to replace it by a partial AUC between 504 hours to 6072. So that's a 3 to 4 weeks. I run the NCA parameter calculation and the bioequivalence, and then I'm going to have the results either as a table or as a plot, where I can see that in this example, all three metrics fall within the BE limits, so we can conclude that the two formulations are bioequivalent. You have seen that it takes only a few minutes to do the entire procedure, but it would be nice to have an automated analysis. And good news, all the steps I have shown you can be done from R using R function that calls the Monolix suite in the background. The R package containing these functions is called Lixoft Connectors, and I can give you a glimpse of it. For the first step, we would first link the R session to Monolix with this line here. Then we would load the Monolix project using load project, and then set the B trial dataset using the function setData, and then estimate the individual parameters using run conditional distribution sampling. I can then go over to Simulix and import the Monolix project into Simulix to predict the carryover, etc., etc. So as you can see, the entire procedure can be done with a few lines of code. This reduced Crawford's design and its analysis procedure looks nice, but does it really work? By does it work, I mean, do we achieve a sufficient power with a reasonable number of individuals and reasonable study duration? And at the same time, do we have a properly controlled type 1 error? I'm speaking of statistical concepts here, so let's do a quick reminder to have everybody on the same page. Bioequivalence analysis is often described as calculating the ratio between the test and reference of the mean of a metric, constructing a 90% confidence interval for it, and then concluding for bioequivalence if the confidence interval is within the BE limit of usually 0.8 to 1.25. But it's also possible to see it as hypothesis testing, where bioequivalence is concluded if the null hypothesis is rejected. And this is done with two one-sided tests at the 5% level of significance. The null hypothesis is that the ratio is either below or above the BE limits. If the null hypothesis is rejected, it means that the ratio is inside the BE limits, and therefore that the two formulations are bioequivalent. The null hypothesis can either be true or false, and the test can conclude that it is either true or false, making a correct or incorrect conclusion. If the null hypothesis is false, so the formulations are bioequivalent, the probability to make the correct conclusion is called the power. If the null hypothesis is true, so the formulations are not bioequivalent, but we conclude they are, we make the wrong conclusion. The probability to make the wrong conclusion is called the type 1 error. For a statistical test, it is desirable to have a high power and to keep the type 1 error under control. The power and type 1 error are probabilities, so they can be calculated empirically over many trials. Here I have shown the results of 10 different trials. Let's assume that the true value of the ratio, if I would calculate it with an infinite number of individuals, is 0.9. Then the correct answer is that the formulations are bioequivalent. For each of the trials, I calculate the confidence interval for the ratio calculated over the individuals of that trial. If the confidence interval is within the BE limits, we conclude that the formulations are bioequivalent, which is the correct answer. For some of the trials, shown in red, the confidence interval is not inside the BE limit, and we take the wrong conclusion. The power is a percentage of correct conclusions. For example, here, with these 10 trials and 8 of them with the correct conclusion that the formulations are bioequivalent, the power would be 80%. The fewer the number of individuals in the trial and the higher the variability, the larger these confidence intervals will be. If the confidence interval is large, it will be harder to be within the BE limit and the power will be lower. We can do exactly the same for the type 1 error. This time we assume that the null hypothesis is true, that is, the formulations are not bioequivalent. So the true ratio needs to be outside of the BE limits. And actually we can put it just at the border here. Most of the trials will have a confidence interval outside of the BE limits, leading to the correct conclusion that there is no bioequivalence. A few of them will be inside. That is a type 1 error calculated as a percentage of wrong answers where bioequivalence is wrongly concluded. Here with these 10 example trials, the type 1 error would be 10% as just one is wrong. With a very large number of trials, we would expect the type 1 error to tend towards 5%. Indeed, the intervals are 90% confidence intervals, which means that the true ratio is outside of the confidence interval in 10% of the cases, 5% on each side. Here we focus only on the lower side, so we expect 5% of the trials to wrongly conclude that the formulations are bioequivalent. For the design and analysis procedures that we have suggested, the power and type 1 error can be assessed via simulations. For the power, we can simulate a large number of BE trials with the proposed reduced crossover design under the H1 alternative hypothesis, so with a true ratio within the interval. Then we apply the analysis procedure and calculate the power as a percentage of simulated trials correctly concluding for bioequivalence. We can vary the number of individuals, we can vary the true ratio. At the end, we hope to have a power as high as possible. And as a reference to compare to, we can do the same procedure for the traditional crossover and for the parallel design. For the type 1 error, we do exactly the same, except that we simulate under the null hypothesis at the limit, so with a true ratio of 0.8 or 1.25. Here the goal is to keep the type 1 error under control, so around 5%. In addition to the routine I have already described, we will thus add an additional step to simulate many BE trial datasets. For this, we will use the model described in the literature, and we can vary the true ratio, vary the duration between the doses, the number of individuals, and we can do many replicates. The clinical trial simulations are Monte Carlo simulations, so the individual parameters will be sampled from the distributions characterized by the population parameters. We will use a 2 single-dose design and realistic measurement times. In order to be able to choose the true ratio value, we will assume that the population parameters for the two formulations are the same, except the relative bioavailability. As the pK is linear, if I set a relative bioavailability to 0.9, for instance, then the expected true ratio for each of my 3 metrics will also be 0.9. So let's try this on our buprenorphine example. There is a published model for the reference formulation that is the following here. It has a fast and a slow route of absorption. You can see the population parameter values here. So I'm gonna apply the routine, and I will first show you the results for a true ratio of 0.95. So that would mean a 5% difference between test and ref, and a 4 months duration between the 2 doses. And here are the results. So in this plot, we see the power versus the number of individuals. If I do a traditional crossover design, it would look like this, and it would take pretty long, 20 months, and the power would be very high. If I go for a parallel design, then it takes only 4 months, but we see that the power is extremely low. With a reduced crossover design as a model-based correction, we have a trial duration of 8 months and a power that is really pretty good with reasonable number of individuals. For instance, we have a 90% power with 30 individuals in the trial. So this is a plot for the partial AUC. I can also check the 2 other metrics. So here we have the plot for the Cmax, AUC last, and partial AUC in case of a 5% difference between the 2 formulations. And I have also tried different durations between the 2 doses, 3, 4, 5, or 6 months that are displayed here. And we see that we achieve a really good power. If instead of a 5% difference, I assume a 10% difference, the power is still very good, except maybe here for the 3 months interdose interval between the 2 doses for the partial AUC. So the power is really satisfying. I also need to check the type 1 error and check that it is properly controlled. If I simulate assuming a true ratio of 0.8, we see that the percentage of wrong conclusions is around 5% for all cases, as expected. If I simulate at the other bound, so with a true ratio of 1.25, We are again around 5% for most of the cases, except for one curve, this one here, which corresponds to the 3 months duration between the two doses of the reduced crossover design. In this case, there seems to be a bias leading to an uncontrolled and unacceptable rate of false conclusions. So to understand what is going on, I have plotted here the B trial data for the first period for a few individuals. When we have data over 6 months before giving the second dose, the individual parameters we estimate on the data, that is the prediction here in orange, are very close to those used for the simulations that are displayed in gray. On the opposite, when the data is only over 3 months, then a fraction of the individuals, for example these two here, do not have data to estimate the decrease of the concentration. And therefore the estimated parameters assume that the concentration will stay very high for very long. And actually the prediction of the carryover into the second period that is too high lead to an overcorrection when we do the subtraction and therefore to a bias. But 4 months is already long enough to get individual parameters that are close enough and therefore avoid any bias. So to conclude, the reduced crossover design with a reference and a test dose 4 months apart allow to have a properly controlled type 1 error and a power above 90% for the 3 metrics with 30 individuals, assuming a 5% difference between test and ref. In this case, the trial duration is 3 times shorter compared to a traditional crossover design with washout, and compared to a parallel design, the power is much, much higher. A nice next step would be to include the dropout rate and check by how much it is reduced if the trial duration is reduced by a factor 3. As a reminder, the procedure is applicable if the PK is linear, if we have a population PK model for the reference formulation, and if the long-acting injectable can be given in healthy volunteers. And we are currently working on a similar method for multi-dose steady-state designs. The advantage of this method is that the individual parameters are estimated for the reference formulation only, for which a population model is available. So there is no real need to check the model validity, although this could be added in addition. And second, the data for the second period is shifted, but we remain really close to the actual data as opposed to other methods based on simulations. The cons is that we do a different processing for the reference and test data because of the one sequence design. The method is implemented as an R script that uses the R package list soft connectors, so it requires a Monolix Suite installation and a license. And the script can be used in 2 ways. For trial planning, to check the power and type 1 error, the inputs to provide would be a Monolix project that defines the model and the population parameters for the reference, and then every information related to the design, so the sampling time, sampling size, duration between the 2 doses, etc. And then we would get as an output the power and type 1 error. Once the actual trial data is available, to do the BE analysis, we would provide the BE trial data and the Monolix project, and we would get as an output the confidence interval for each metric and the BE conclusion. So the reduced crossover design provides good power, reasonable study duration, and a controlled type 1 error. It is implemented as an R script using the Monolix Suite. Thank you very much.
Hi, my name is Parmesh Gajjar, and I'm a principal scientist at Seda Pharmaceutical Development Services. I'm delighted to be able to present our work today on a model integrated pathway to explore the bioequivalence of long-acting injectable products. We've heard a lot today about how long-acting injectable products have become a popular choice to treat a number of conditions. This graphic also shows that the number of generic products that have been able to penetrate onto the market are very few. At the same time, mathematical models have become a popular choice in a number of submissions in the last few years. Can these mathematical models be used to aid the bioequivalent process. In particular, today I'd like to explore how population PK modeling can be used to identify which products are bioequivalent after multiple dosing, to determine whether we need multiple dosing, and to ask how risk can be reduced in bioequivalent testing through the use of modeling. What I'd like to present is deliberately radical in order to drive and stimulate new thinking. As an example, I'd like to use paliperidone palmitate, as this has a well-established population PK model in the literature. However, everything that I will present could equally be extended to other long-acting injectable formulations. The model I will use is that from Samtani et al. From 2009, and that's the basis of this work. A starting question is, what is a population PK model? A PK model is a mathematical structure to predict PK profiles by representing the distribution of the drug through different virtual compartments. For example, this is the PK model for paliperidone palmitate. The model takes into account the volume of distribution, the clearance, and the absorption rate of the drug. Long-acting injectable products often have different fractions of the full dose inputting at different rates. We can understand this by looking at the release profile, where we see that this particular model gives us a fraction F2 that enters very quickly, with the remaining portion of the dose entering much more slowly after a time t lag, when it is absorbed at a rate of Ka. Now, the PK model allows us to simulate different PK profiles, such as the profiles for 100 mg and 150 mg doses that have multiple doses administered. A population PK model develops this further by including between subject variability. For example, in this particular model, the volume is dependent on the individual's gender and BMI. The clearance is dependent on the individual's creatinine clearance. The absorption rate depends on the individual's gender, injection site, and age. And the dose fraction depends on the individual's gender, injection site, needle length, and injection volume. This allows us to simulate unique PK profiles for each individual. For example, here are 25 virtual individuals based on the US population who have been given multiple doses of the reference product. It's usually easy to represent these multiple individual profiles using a geometric mean, shown here with the solid black line, and a 95% confidence interval, shown with the grey ribbon. For the remainder of this talk, we'll represent population PK profiles by such a geometric mean and a confidence interval. However, it should be understood that individual profiles are wrapped up within this confidence interval ribbon. The first question we wish to answer is, how can we identify bioequivalent products after multiple dosing? We'll use our population PK model to simulate PK profiles of different products after multiple dosing. We'll compare the performance of these products using appropriate metrics, and we'll determine which products are bioequivalent. The beauty of a mathematical model is that we can vary different parameters effortlessly. For example, here we have 5 different parameters, which we can change. Let's take a product, test 1, which has 80% of the absorption rate of our reference product. We can simulate a PK profile for this. Let us also take a second product, test 2, which has 50% more of the dose input during the fast phase. We wish for a way to compare these different products. The standard metrics that are used are Cmax, Cmin, and AUC, which we can calculate from our virtual profiles. In addition, in recent years, there has been a call for introduction of partial AUCs, and we'll consider 3 of these as well. AUC from 0 to 3 days, AUC from 0 to 28 days, and AUC for a steady state profile. We can calculate each of those metrics for each product, and we can calculate a ratio between the reference and the test product. Now, if the ratio lies between 0.8 and 1.25, then the product is considered to be bioequivalent. For example, here, test 1 is bioequivalent. However, if any of the metrics fall outside of that range, then the product is not bioequivalent. So test 2 is not bioequivalent because it fails on Cmax, AUC from 0 to 3 days, and AUC from 0 to 28 days. To make our simulations a bit more tractable, we can make a number of simplifying assumptions. The first assumption that we'll make is that the volume and clearance are properties of the drug, i.e. do not vary from formulation to formulation. All the other parameters shown in green are considered to be properties of the formulation. So we'll vary the dose fraction F2, the time lag, and the absorption rate to simulate different products. After simulating different products, we can then calculate the ratios of the different metrics and determine if those products are bioequivalent or not. For each metric, we can consider it by itself. So for instance, if we consider Cmax, we can see that the light region are products where Cmax ratio is below 0.8 of the reference, and the dark region is where the Cmax ratio to the reference is above 1.25. The remaining space, shown in white, are the bioequivalent products. We can perform a similar analysis for all of the other metrics, Cmin and AUC, and putting them together, we gather a formulation parameter space, of which the white region are bioequivalent products. We can highlight these specific products in blue here, and these are the products that are bioequivalent based on Cmax, Cmin, and AUC. We can also repeat this analysis including the partial AUCs, and this gives us a new formulation parameter space. As we notice here, the space is much smaller. Again, if I highlight these products specifically, we can see that it is a small region. And comparing this to those products that we found were bioequivalent using Cmax, Cmin, and AUC, we can see that the use of partial AUCs restricts the number of bioequivalent products. This ability to examine the influence of different metrics is very important. And along with the PK profiles, this can be used on a case-by-case basis to determine which are the appropriate metrics for the product at hand. At the start, I said there were 3 parameters that I were varying: Ka, Tlag, and F2. So this is in fact a 3-dimensional parameter space. But if we look at this in 3D, we see that there is some relationship between Tlag and F2. Now, this indeed looks like a proportional relationship. So for simplicity, we'll assume that we just look at a value of Tlag at 100% and simply vary the dose fraction F2 and the absorption rate Ka. Looking back at our formulation parameter space, we can consider the previous profiles of test 1 and test 2. Test 1 was bioequivalent, and we can see that it lies within the formulation parameter space for bioequivalent products. On the same note, test 2 was not bioequivalent and lies outside of this region. And looking at the individual profiles, we can see that the large dose fraction that enters in test 2 means that this fails on Cmax and AUC. So to summarize this section, we can see that population PK modeling has allowed us to simulate different PK profiles for different products. We can see how the choice of bioequivalent metrics affects which products are considered bioequivalent. And we've discovered a range of products, a formulation parameter space, which are bioequivalent after multiple doses. The next question that arises is, do we need multiple doses? We can simulate the PK profile of different products after a single dose, and we can compare them using appropriate metrics. But now we wish to determine which criteria recovers the multiple dose bioequivalent products that we found previously. So as per the multiple doses, we can simulate the PK profile after a single administration on day 1. And in a similar fashion, we can calculate the different metrics. Here, the only partial AUC that is relevant is the AUC from 0 to 3 days. Normally, we look at the bioequivalent ratio between 0.8 and 1.25. However, we wish to ask an inverse question. Which bioequivalent ratio gives us the formulation parameter space of bioequivalent products? Is it between 0.8 and 1.2, which I will call a delta of 0.2? Is it between 0.9 and 1.1, a delta of 0.1? Indeed, we can group all of the products by which delta all of the metrics fall under. This creates a very colorful parameter space. The darkest color represents the smallest delta, very close to the reference product at 100, 100. And as we move further and further away, there is much greater variability in the different metrics. We wish to find which delta recovers the bioequivalent products that we found after multiple dosing. And indeed, by setting a delta of 0.2, we can recover those products. Here, the multiple dose bioequivalent products are shown in red, with the region that we just found with a delta of 0.2 shown in black. If we examine the delta groupings for each individual metric, it is possible to find a different mapping, a mapping that includes more of the products. A simple extension shows that we can find an individual metric balance that gives the yellow space a much wider space of parameters. And so by determining individual ranges on Cmax, Cmin, AUC, and the partial AUC from 0 to 3 days, we will be able to recover the full space of bioequivalent products after multiple dosing. So what we've managed to determine is that we can use population PK modeling to determine a criteria on single dose studies that gives the products which are multiple dose bioequivalent. Now, you've heard me talk for 15 to 20 minutes already, and many of you will be asking, how does the modeling integrate into product development and testing? The first aspect is that it can guide product development. We can take our formulation parameter space of bioequivalent products, and we have a target for our engineering. We can attempt to engineer a product that has a release profile that lies within the range of bioequivalent profiles. However, I do not wish to concentrate on this at the present time. Instead, I'd like to present some ideas on how modeling can integrate with bioequivalent assessment. A normal bioequivalent study runs as follows. We obtain PK profiles from N individuals for the reference product, and we obtain PK profiles for N individuals for the test product. We calculate metrics, and we compare the metric ratios. And if they lie within our determined range, they're bioequivalent, and if not, we reformulate or we stop. Here is some virtual PK data based on a virtual population of 50 individuals based on the US Census. I'd like to apply this method to real data and would be happy to work with anybody that has that same interest. With this virtual population, I've conducted a virtual crossover study where each individual has received single doses of the reference and test 1 product. Now, there are individual profiles, for example, shown here for individual 5 and individual 22, where they've received the reference and the test product. For each of these individual profiles, we can calculate the appropriate metrics. For instance, Cmax, Cmin, AUC, and AUC from 0 to 3 days. From this, we can calculate a ratio. And taking all of the individuals into account, we have a ratio for the entire product. We can determine whether this lies within our range and determine if it's bioequivalent or not. However, there's a lot of risk in this process. There's a need for each individual to take 2 products, and this means that our study lengths can be very long. And there's also high inter-individual and inter-occasion variability. Now, this leads to a lot of risk. Could we reduce that risk by eliminating the need for the individuals to take the reference product? Here is one idea for a model integrated bioequivalent study. Here we obtain the PK profiles for n individuals who only receive the test product. To those individual PK profiles, we fit a population PK model, allowing the formulation parameters to vary, and we determine if that product is model bioequivalent, i.e., does that lie within the formulation parameter space of bioequivalent products? If yes, then that product is bioequivalent, and if not, then we reformulate, or we have the option of conducting a full bioequivalent study. So if we take the individual PK profiles, we can fit a population PK model to each of the individual profiles, allowing the different formulation parameters to vary. This gives us a set of model parameters, but it's not the individual values by themselves that are important, but their percentage of the reference product. So here, the absorption rate is 79% of the reference product, and the dose fraction F2 is 107% of the reference product. We can map this on the formulation parameter space, shown here with a cross marker. At the same time, we had the values for the test one product, and so we can see in closeness how the two compare. This shows in itself the variability from PK studies. This work was done using point ratios. However, we can also fit a confidence interval and ask that the confidence interval lies within our formulation parameter space. Similarly, we can look at the values obtained by fitting models to virtual PK studies of the reference product and test 2 products. And as expected, we can see that reference and test 1 lie within the formulation parameter space, and test 2, which we determined to be not bioequivalent, lies outside of this space. This approach can also work if we do not know the formulation parameters a priori, as will happen with real products. Let's take a product test 3, which we do not know what its formulation parameters are. We can fit a population PK model to this. And examining that on the formulation parameter space, shown here with the yellow marker, shows that it is a bioequivalent product. This has a number of advantages over a traditional bioequivalent method. It removes the need to trial both the reference and the test products, making clinical trials more streamlined. And the use of models allows for both inter-individual and inter-occasion variability to be accounted for. Thus, it's a very powerful approach, with potential not only for the other long-acting injectable products with models, but also for other complex parenterals, where it is a challenge to demonstrate bioequivalence. To conclude, we've managed to use population PK modeling to determine a range of products which are bioequivalent after multiple dosing. We've determined what criteria we can use to recover bioequivalent products after only a single dose. And we can create an integrated process with a PK study to prove bioequivalence with lower inherent risk. I'd like to finish by thanking all of the team here at SADA who have helped to put all of this work together.
Thank you, everybody, speakers and participants. That we will now close the morning session and we'll have a lunch break until 1 p.m. So please come back to the program by 1 p.m. Thank you. See you soon.